All right, what if we could actually make a living thing immortal? I'm not talking about some sci-fi movie. I'm talking about doing it in a real lab. Well, that's the wild question that sent one researcher on this incredible journey deep into the science of aging. And their subject? Something you probably have in your kitchen right now: yeast. And you gotta understand, the stakes here are just massive. We're not just talking about adding a few more years to a life. We're talking about stopping the aging process cold. Like the researcher said, if we could pull this off, it would literally be the first time humanity has ever made an organism immortal. I mean, that's a huge deal for our understanding of life itself. And it really all boiled down to this one kind of simple sounding question. Let's forget about humans for a second. Could we just take regular old baker's yeast and somehow engineer it so it just doesn't age? That one question, that's what sparked this intense week-long deep dive right into the heart of biology. So, okay, you're a scientist and you've got this massive question in front of you. Where do you even start? I mean, you can't just run to the lab and start mixing things. No, you have to have a process, a way to really get your head around the problem first. So let's break down exactly how they did it. And it's basically a 3-step plan. First step, you've got to build a mental model. You really have to get inside the head of a yeast cell. Step 2, you hit the books, or, the scientific papers, and see what everyone else has already figured out. And then step 3, you start challenging all of it. You ask the hard questions and look for the gaps. And I gotta say, this first step is so cool because it's not about numbers or data, it's about empathy. The researchers said they literally tried to imagine, what would I worry about if I were a yeast cell getting older and bigger? Things like, am I getting enough power? It's this amazing way to build a gut feeling for the problem before you just drown in all the details. Okay, so you've got your mental model, you've done your homework, and that's when this simple, elegant question about making yeast immortal just slams face first into a giant wall. A wall of just insane complexity. What's the wall made of? Well, it's made of this number, 49. That's right, 49 different documented biological changes, what scientists call hallmarks, that are all tied to yeast aging. So this isn't about finding one thing that goes wrong. It's about a total system failure, a chaotic breakdown happening on like dozens of fronts all at once. Okay, let's just rapid fire through a few of these so you can get a feel for it. First up, the cell size just increases. The whole thing just gets bigger as it gets old, a pretty clear sign that things inside are getting out of whack. And then inside, the pH in its little recycling center, devacuole, it starts to drop. It gets more acidic, which means it can't clean up cellular junk anymore. So garbage just starts piling up. Next, you've got the mitochondria. The power plants of the cell. Well, they start to break apart. They fragment. So boom, you've got a massive energy crisis on your hands. And it doesn't stop there. Its ability to even process sugar for energy, a process called glycolysis, that starts to tank too. So the cell is basically starving itself from the inside. And even a key part of the nucleus, the nucleolus, starts to swell up. Look, the point isn't to memorize all this stuff. The point is to feel the weight of it. How on earth do you even start to fix a problem that's breaking in 49 different ways at the same time? So, you're totally buried under this mountain of 49 different problems. It feels hopeless, right? But it turns out, hidden in all that same research, there was a little glimmer of hope. It looked like a potential biological reset button. And this process is called sporulation. You can think of it as a yeast cell's emergency escape plan. When things get really bad, it can basically bundle up its DNA into these tiny, super tough spores. And these spores, they're essentially younger, fresh versions of the original cell. But of course, it's not a perfect fountain of youth. Turns out, an old, tired yeast cell is way worse at this trick. Its ability to do this, its sporulation efficiency, it just plummets from almost 70% all the way down to under 17%. And that leads to a huge question. Is this process really reversing aging? Or is it just that only the healthiest, strongest old cells can manage to do it at all? And this is where the whole journey takes a really crucial turn. Because finding a potential hack like this sporulation thing, it just wasn't enough. For a real scientist, a trick isn't the answer. The real prize is understanding why the trick works. This quote from the researcher just nails the scientific mindset perfectly. The frustration isn't that the problem is hard, it's that the answers we have feel kind of shallow. It's this deep need to go beyond just finding what works and to truly get why it works, right down to the fundamental physics of it. And this really highlights two totally different ways of doing science. On one hand, you have the genetic screen. That's basically a brute force attack. You just test every single gene in the yeast genome and see what sticks. But what our researcher really wants is the second approach, a physical model. A short list of smart, testable ideas. Think of it like this. One way is trying every single combination on a lock. The other way is understanding how the lock actually works. The researcher even called this huge gap in our knowledge uncharacterized dark matter. And that's such a perfect way to put it, right? It means there are probably some really basic, fundamental rules about aging that we don't even know exist yet. And until we find them, we're just kind of stumbling around in the dark, treating symptoms instead of the real cause. And this leads to a moment of just great, grounded humor. The researcher basically says, look, what I'd really love is to have tiny little nanobots that I can just send in to move atoms around wherever I want. But then they add, I guess the universe doesn't really care about my desires. It's just a funny, honest way of saying, hey, we can only work with the tools we actually have. So after this whole week-long whirlwind, what's the big takeaway? It's this. The mission to make an immortal organism isn't really about finding one magic gene or some clever little trick. The real challenge, the fundamental challenge, is getting a deep physical understanding of why a complex machine like a cell breaks down to begin with. And that leaves us with a really big question, not just about yeast, but about science itself. When we face huge challenges like aging, what's the best way forward? Is it to just throw everything at the wall, screen every single gene and see what works? Or do we need to slow down, take a step back, and really focus on building a deeper, more fundamental understanding of how life works first? Honestly, the answer to that question could shape the next 100 years of biology.